Hello, and welcome to our presentation on human myeloid-derived growth factor, abbreviated MyDGF. To start right off, we're going to give a bit of history regarding this protein. You can see here in figure 1 the general structure of MyDGF as determined by X-ray diffraction experiments. The N terminus is colored blue here, while the C terminus is red. Now, this is a very unique structure for a protein, which we'll get into. What I will mention here is that the earliest papers we were able to find that went into sufficient depth on MyDGEF were as recent as 2018. Since then, while significant discoveries have been made, it is true that in 2018, even the structure of MyDGF was unknown. While there are still many unknowns, now 4 years later, a lot has been discovered, which we will describe. So a bit of history of the discoveries of MyDGF. As I had mentioned in 2018, both the structure and function were unknown. The structure problem was solved in 2019 by NMR at the University of Wisconsin-Madison. To do this, they had to grow the protein in NMR active isotopes, nitrogen-15 and carbon-13. And due to the differing isotopes, they had to ensure that this didn't affect the protein folding. Fortunately, as verified by mass spectroscopy, the proteins were found to be identical, so they could proceed with the NMR experiments. What made these NMR experiments possible was the recently published crystal structure of a KDEL receptor in 2019, shown here in figure 2, which allowed for computational docking of MyDGF onto this receptor, which we will explain more about shortly, which is why this was able to work. The NMR experiments were carried out in solutions near physiological conditions at the University of Wisconsin-Madison and have continued at Oak Ridge National Laboratory. With these experiments, they found that a base domain of an enzyme called vannin adopts a similar fold as MyDGF, and as a result, in 2020, a new protein family was made, UPF0556 or PF10572. And this was created by Alex Bateman of the European Molecular Biology Laboratory and the European Bioinformatics Institute that encompasses those sequences related to MyDGF protein and vannin-based modules. This protein family is largely composed of the MyDGF proteins across the various species. However, it still holds that MyDGF has an incredibly unique structure and does not fit any other protein fold class known today. Interestingly, this protein actually exists in nearly every tissue and cell in the human body and across hundreds of different species, and so it is evolutionarily conserved. Given that, while more research is certainly needed to determine its specific function and binding partners, of which both aren't clearly understood at this time, it will be interesting to discover what evolutionary function it has served and across so many species. So with the structure determined, more research has gone into how MyDGF functions, and much has been discovered. It was found that MyDGF is secreted from monocytes and macrophages, specifically CCR2 high monocytes in macrophages and CCR2 low macrophages in the left ventricle, which are known for their pro-inflammatory and reparative functions, respectively. MyDGF is also retained within the ER and Golgi body and participates in paracrine-like signaling, functioning in many inflammatory processes from acute MI to ischemic stroke to type 2 diabetes. So Milena will now describe the details of this unique protein structure and subsequent function.
Okay, so for the structure of MyDGF, here are a few things that are notable to mention. Well, as mentioned before, MyDGF is paracrine acting, and paracrine signaling is a type of cellular communication in which a cell produces a signal to induce changes in nearby cells, altering the behavior of those cells. MyDGF itself is a protein that is produced by bone marrow-derived monocytes and macrophages, which are responsible for the protection and repair of the heart after MI. Another thing that will be mentioned are epitopes, and these are part of antigen molecules to which antibodies attach themselves to, and this will be significant since the X-ray structure of MyDGF suggests and identifies an epitope that is functionally relevant in terms of binding to a receptor. MyDGF is also a 10-stranded beta sandwich with a folding topology, to which I will also explain in more depth as we go along the presentation. In general, in terms of putting this protein into a certain class of families, ultimately scientists were pretty much unable to. However, MyDGF does share similarities to other beta sandwich proteins, such as members of the immunoglobulin and jelly roll fold, or the beta gamma crystalline superfamily. But it also has certain single strands and a different number of strands, which makes it different from all other known protein classes. Another thing to note is that there are 3 surface patches that are mutated in the 3 inactive MyDGF variants that are located in close proximity to each other, and they are located near MyDGF's top face face on loops 5 and 7. These MyDGF patch mutants were designed to repulse possible receptor binding due to the inversion of charges. As mentioned before, MyDGF is a 10-stranded beta sandwich with a folding topology, meaning that it consists of 10 anti-parallel beta strands, forming 2 beta sheets, which are packed face-to-face to each other, forming a beta sandwich. The beta strands are connected by loops of 3 to 18 amino acid lengths, enclosing a hydrophobic interior, and the loop structure itself is asymmetric. Also present are short hairpin-like loops located on one end of the beta sheets, while 3 elongated loops are on the other end. The area holding the elongated loops is known as the top face, and the area with the shorter loops is known as the bottom face. The important part really, loops 5 and 7 are the top face most prominently elongated loops. These loops are also within a lateral position and protrude in a 90-degree angle over the core of the beta sandwich. The beta sheet plane that has beta strands 1, 4, 5, 10, and 7 located on the side where the loops are projecting is called the front face. But the opposite beta sheet with beta strands 2, 3, 6, 9, and 8 is called the back face. Also, a single disulfide bond between cysteine residues 32 and 61 of beta strands 3 and 5 connects these opposing beta sheets. There is an applied dimer there with a buried surface area of 1,726 angstroms, which seems to be the relevant form. And from this, they found this dimer consists of monomers and concluded that MyDGF is a monomeric cytokine. MyDGF, like I said, has patch mutants, and this implies that the mutated residues are directly involved in the receptor interaction by limiting binding. Human MyDGF has 142 amino acids and consists of an N-terminal 31 residue long signal peptide, and the MyDGF protein sequence contains a C-terminal KDEL-like ER retention sequence. With this information, scientists were able to localize the receptor interaction interface to a region around 2 surface tyrosine residues, which were 71 and 73, and an adjacent prominent loop structure consisting of residues 97 to 101. Next, we're going to talk about Fab8, and Fab8 was one of the potential components that scientists thought participated in the binding affinity of MyDGF. However, scientists eventually then found out that Fab8 actually blocks receptor binding, and since binding of Fab8 blocks receptor binding, which leads to the neutralizing of MyDGF's biological activity, the asymmetrically localized epitope implies that the surface regions of MyDGF that are positioned opposite to the epitope aren't relevant receptor recognition sites. So even though Fab8 interferes with binding, scientists were still able to at least identify the areas that are not relevant receptor recognition sites. Residues tyrosine 71 and tyrosine 73 were assumed to directly interact with the receptor, and most probably are key residues that control receptor binding affinity. Tyrosine 71 is identical across all species, while tyrosine 73 is highly conserved and replaced by the aromatic amino acid phenylalanine in non-mammalian sequences. Tyrosine side chains are often involved in protein-to-protein interactions. The high proportion of aromatic residues in some tyrosine and tryptophan included in protein-to-protein interactions reflects a general property of antibody epitopes. It is, however, important to note that also mutations which are located on loops 3 and 9, adjacent to tyrosine 71 and 73, have no effect on MyDGF's activity. However, as a whole, this observation suggested that the receptor interaction is more likely to take place in the front protruding part of the top face, and indicates that tyrosine 73 may be the key residue for the actual receptor interaction. One last thing to note for the structure is that a receptor that is normally associated with MyDGF is CXCR4, and it is a chemokine receptor that seems to affect the size of an infarct. And while I couldn't find too much detail on this receptor, it's important to be aware that the presence of this receptor affects how much damage occurs to the heart, and therefore it could even be hypothesized that MyDGF utilizes CXCR4 when myocardial infarction takes place so that much damage isn't done to the heart. Now let's move on to the function. In terms of function, MyDGF showed improvement in wound healing and cell migration, which seems similar to treatment involving vascular endothelial growth factor. Scientists observed MyDGF inducing endothelial cell migration, and this observation led to the functional implication in the identification of the relevant surface epitope of MyDGF that is part of the generally considered receptor binding site, which is key to its biological function. MyDGF has also shown to improve tissue repair and heart function in mice. And to go more in depth about that, while comparing to wild-type mice, it was found that MyDGF deficient mice developed larger infarct scars and more severe contractile dysfunctions. Therefore, treatment with recombinant MyDGF helped protect and repair the heart after acute MI. MyDGF was therefore seen to mediate cardiac repair after MI takes place by inhibiting cardiac myocyte apoptosis to reduce the size of the infarct. Another thing to add that was also done by scientists was that loss of function and gain of function experiments in mice revealed that MyDGF also induces glucagon-like peptide 1 production and secretion in order to regulate glucose levels, which improves glucose tolerance and lipid metabolism, which Katie will explain how this would be relevant in terms of diabetes. In terms of inflammation, transgenic overexpression of MyDGF in bone marrow-derived inflammatory cells were found to reduce pressure overload and induce hypertrophy and dysfunction. In general, the adult mammalian heart has a hard time repairing itself after events such as MI. Cardiomyocyte necrosis, also known as body tissue death, triggers an intense inflammatory reaction leading to the formation of collagen-based scar. When this happens, the infarcted myocardium is then infiltrated with abundant myeloid cells that interact with cardiomyocytes. Therefore, these observations suggest that the myeloid cells secrete paracrine factors that may be responsible for pathological remodeling, inducing generation of new blood vessels, and even myocardial regeneration. Once again, CXCR4, a chemokine receptor associated with MyDGF, affected the size of an infarct, and they saw this in mice, where the mobilization of CXCR4 bone marrow-derived cells reduced infarct size and improved cardiac function after MI. Well, now that we know something about the structure and function of MyDGF, how are the two connected? In general, Old Paper's research implied that not much is known about MyDGF structurally or functionally, but there are a few key takeaways. The observation considering the binding receptor once again suggests that the receptor interaction is more likely to take place in the front part of the top face, which implies once again that tyrosine 73 is most likely the residue for a receptor interaction to take place. With this information, these findings are able to at least provide a basis for structure-guided protein engineering in order to eventually develop modified MyDGF variants, which could potentially become advanced therapeutics for things such as MI when in clinical use. To reiterate, MyDGF being abundantly present in the left ventricular myocardium and in blood plasma of pressure overloaded mice showed regenerative properties involving the heart, which normally has little regenerative properties. Therefore, these findings also implied a MyDGF-based adaptive signaling between inflammatory cells and cardiomyocytes that protect against pressure overload-induced heart failure. In an acute heart attack, there is a disruption of blood flow causing myocardial cell death, and this damage to the myocardium leads to inflammatory cells becoming necrotic, eventually leading to a collagen-rich scar formation that interferes with heart function. In most cases, that involves a significantly large MI where massive myocardial loss cannot be prevented, but with the emergence of bone marrow cell transfusion, a type of therapy that involves MyDGF, there is a possibility to salvage oxygen-deprived heart tissue and reduce scar formation. Okay, so since I gave you definitely the microscopic things, let's give it back to Katie to tell you more about the big picture stuff.
So yeah, what do we know, big picture-wise, when MyDGF doesn't function correctly or is completely absent from the body? Well, the primary discoveries of this protein involve acute myocardial infarction or heart attacks. So MyDGF was found to be an angiogenic growth factor, meaning it helps in the development of growth and new blood vessels, but also appears to play an inflammatory role as well. How is inflammation related to heart attacks? Well, specifically with the more detrimental effects of heart attacks, namely heart failure, chronic inflammation plays a crucial role in that pathogenesis. For example, cytokines and growth factors in the myocardium, the major muscular tissue of the heart, will promote contractile dysfunction and adverse ventricular remodeling through their interactions with parenchymal, and stromal cells within the ventricles of the heart. And yet, these are negative implications of the immune system that progress heart disease. However, in cases where macrophages were completely absent, there's a complete decline in functionality. So the immune system is still necessary to preserve the functionality of the heart after events such as a heart attack. In a recent study, scientists subjected wild-type mice and MyDGF knockout mice to acute pressure overload of the heart, which is known to induce an inflammatory response in the heart. What was found was that those mice that did not have MyDGF had a higher percentage of cardiac hypertrophy, fibrosis, contractile dysfunction, and impaired calcium cycling and sarcomere functioning compared to wild types. They determined that MyDGF actually stimulated SERCA2A or sarcoendoplasmic reticulum's calcium ATPase2A expression in cardiomyocytes, thus aiding the heart in its normal function, protecting it against heart failure development. Interestingly, the wild type MYDGF knockout mice did not appear different prior to the acute pressure overload treatment, and there were no developmental differences or dysfunction previously. And the MYDGF knockout mice appeared perfectly healthy. It wasn't until after the inflammatory response was triggered that researchers were able to tell a difference. So this slide shows a few figures regarding MYDGF's role in attenuating left ventricular remodeling after acute pressure overload. Figure H, which I've highlighted here, specifically gives a great comparison between the recorded pressure within the left ventricle between MYDGF wild type and knockout mice after undergoing a sham surgery or the TAC surgery, which induces acute pressure overload. You can see that the pressure in the KO mice is much higher than the pressure in the wild type mice after TAC surgery, indicating the presence of MYDGF is able to reduce the pressure experienced within the left ventricle. This slide shows MYDGF expression in wild type and KO mice during pressure overload. I've highlighted figures B and C in particular. B shows the relative levels of immune cells in the left ventricle after TAC surgery, showing that there is by far a higher percentage of macrophages. Figure C shows the mRNA expression of MYDGF in these various immune cells, showing that MYDGF is present in the highest concentrations in monocytes and macrophages. These two graphs taken together show that after acute pressure overload, MYDGF, through its elevated expression in macrophages, is present in high quantities. Finally, this last slide shows a comparison between wild type and knockout mice regarding calcium cycling, sarcomere function, and SERCA2A transfer. I've emphasized figures E, F, and G here. Figure E specifically shows sarcomere contraction amplitude and is observed to be higher in wild type mice after TAC surgery. This indicates improved functioning compared to the knockout condition. As for figures F and G, they show maximum systolic shortening velocity and maximum diastolic relaxation velocity, respectively. In both scenarios, wild type mice have elevated velocities, thus showing improved functionality of the heart after pressure overload. Overall, this shows MYDGF's likely role in maintaining the functionality of the heart after acute pressure overload, thus mitigating the effects of heart failure. But the applications don't end there. MYDGF may also have applications within the pancreas and type 2 diabetes. Specifically, MYDGF deals with the incretin system, which is responsible for controlling the glucose-induced insulin secretion, and also includes hormones such as glucagon-like peptide 1, GLP-1, and glucose-dependent insulinotropic polypeptide, GIP. Those two hormones in particular are important in the promotion of the growth and development of beta cells in the pancreas, thus preventing their decay via apoptosis, and as we know, beta cells are what secrete insulin. For those with type 2 diabetes, not only do they have an inability to take up glucose, but their incretin system that I've described here is severely reduced or completely absent. So GLP-1 and GIP are not being secreted, and therefore insulin secretion is impaired, and we end up with the disease process of diabetes. MYDGF specifically appears to promote the production and secretion of GLP-1. And so it may be a therapeutic target for those with type 2 diabetes and may aid in insulin secretion, even if the incretin system itself is non-functional. This research, though, is still new, and more is needed to completely understand MYDGF's role in diabetes. But ultimately, the one common denominator is that MYDGF is involved in inflammatory processes. It is possible that MYDGF could aid in disease processes such as an ischemic stroke and inflammatory joint disease, as both include insufficient perfusion and thrombotic occlusions. And of course, with both, an inflammatory response is triggered. At this point, once again, more research is needed to determine MYDGF's role in these processes. So in conclusion, MYDGF, while evolutionary conserved, has only recently been discovered in terms of its structure, its specific structure and function. Research into both, however, has been difficult due to the structure of MYDGF being incomparable to any other known protein fold class cytokine or growth factor, so much so that a new protein family was created for it. While studies have shown MYDGF's role in inflammatory processes, binding partners of this protein and its active site are unconfirmed at this time. Additionally, while many studies have shown what happened to MYDGF when it's not present at all, studies involving a mutated MYDGF or small molecule inhibition of the protein have not yet produced results. Nevertheless, it is certainly exciting to catch a protein during this discovery stage and see the developments of the process determining its structure and function. I know for Milena and I, following along in the recent discoveries has been fascinating, as the implications of such a unique protein unfold before our eyes.
Yeah, it really has been quite the journey in terms of dealing with this protein since not much is known about this. So let me just remind you guys what seems to be pretty solid so far. So just to reiterate, tyrosine 73 is most likely the residue for receptor interaction to take place. And knowing this really provides a basis for structure-guided protein engineering so that we can eventually develop therapeutics for sufferers of MI. Once again, while MyDGF doesn't exactly fit any protein class, it still shares similar characteristics with other beta sandwich proteins, and the differences being seen are most likely due to evolutionary reasons involving the protein. Finally, once again, the emergence of BMC transfusion is something that is very promising for sufferers of MI, and MyDGF is the promising protein that could potentially even further BMC efforts. Therefore, I can tell you our protein definitely feels mysterious, but so far, like Katie has said, it's unique. And it seems like as the years go by, hopefully when someone else decides to do a presentation on MyDGF, it's no longer a mystery.
Yeah, so these are our sources, and thank you so much for watching.
Thank you.